Well, hey everyone, hey Anatomy. Hope you're doing well when you watch this video. Hope you and your family are enjoying each other. Keep praying, keep hoping and trusting in God. We're going to make it through this weird, uncertain time, one day at a time and together. So today I hope to help you understand adaptive immunity. It's different than non-specific or innate immunity that we talked about in a previous video. And so you can kind of read over the objectives right here. But basically my goal is to help you understand adaptive immunity, how antigens kind of trigger a response, and how antibodies work, what they are, and how they work. Okay. And so today, specific immunity, it's also called adaptive immunity. It's our ability to recognize, respond to, and remember harmful substances. There's two different types that you should be aware of. There's natural immunity and artificial immunity, okay? So natural immunity, it's not deliberate. Like you don't get anything injected into you deliberately. It just kind of naturally happens. So for example, you get a disease, an active disease. So people who have had COVID-19, their body has produced antibodies. That's active natural immunity. And eventually, we're talking about that herd immunity, right? And so that is an example. Passive immunity, passive from the mother to the fetus through placenta or the breast milk. Okay, mother's milk, from mother to child. Artificial immunity, well, this is where active would be vaccination. That would be considered active. Passive would be if protective material developed in another individual's immune system and is given to a non-immune individual. This is where the antibodies come in. When they're doing these plasma tests and antibodies present in plasma and giving that to people who are sick to protect them. So that would be passive artificial immunity. You need to know the difference between natural and artificial specific immunity. So adaptive immunity is coordinated and produced by T cells and B cells. It's not present at birth. It's acquired by either an exposure to an antigen, which would be considered active, or receiving antibodies, which would be, oh, don't want to drag it, which would be considered passive. So active versus passive, okay? So here's a helpful chart that I already showed you once before, but this chart is helpful from a previous anatomy book I used elsewhere, I guess. But non-specific versus specific, I would just kind of know this chart pretty well, okay? Or at least know where to find it and to review it, because this is where we're really kind of understanding specific versus non-specific. We will talk about complement again today because complement does help with antibodies, but complements are mostly considered in the non-specific system. And so here is types of specific immunity: natural, active, and passive; artificial, active, and passive. Okay, so again, this table right here would be helpful to go under. So vaccination. I don't know if you've ever met someone who's anti-vax. They're not for vaccination. They're against it for whatever reason that is. There's been some link, some reports that it increases mental learning disability and various things like that. That's been debunked. And there's really interesting videos you can kind of learn about why people are anti-vaccination and what happens. But I want you want to do what's best for your kids. And so, when my oldest girl was, during that time, I didn't really struggle with it because I, trust in vaccines. Vaccines have saved more lives than anything else in the history of the world. But vaccination, it was still, like, there is that little tinge of, well, what if? But you just give it to Jesus and you move on. But vaccination is, and I'll put some of these links in the video box. But vaccination, this one right here will be very helpful. Great cartoon, explains how vaccines work. I would like for you to look over that one, and some videos about vaccination. But vaccination is very important. You can see here that pertussis, causes various diseases. It kind of was really low, right? And then all of a sudden it's kind of making its comeback. Well, why is that? Possibly, I mean, possibly because of anti-vaccination. This is from the CDC website, anti-vaccination, people against vaccinations. In 2019, there was 1,282 individual cases of measles. That's like crazy high since 1992. And so why did this happen right here? The majority of cases were among people who were not vaccinated against the measles. And so these deadly diseases that we hear about, most doctors who study all this, they're given a specific name. I can't remember it right now, epidemiologists maybe. But these guys really are, yeah, these guys are really, really, proponents of vaccination, of course, because vaccination protects diseases from infecting us. And so these diseases are still around. It's not like they've been completely wiped off the planet. It's just that vaccinations have reduced their incidence among the population. But however, if people start becoming unvaccinated, these diseases could make a comeback. So questions part one is dealing with um, vaccination. So I want you to explain the process. How does it help the body remember and defend against a pathogen? What is the update for the vaccine to defend against COVID-19? So I want you to do a little research and tell me what the status is for that vaccine for COVID-19. Okay, so properties of the adaptive immune system. First off, specificity. T cells and B cells have receptors for only one specific type of antigen. So responses of activated T or B cells are also specific. So they know exactly what they're targeting. Versatility. Millions of lymphocytes, each sensitive to a different antigen. You can see the millions of different combinations here. When activated, a lymphocyte divides, produces more lymphocytes with the same specificity. So all cells produced by the division of an activated lymphocyte constitute a clone. Third property is memory. Activated lymphocytes produce two groups of cells, groups that attack invaders immediately, and then groups that remain inactive unless exposed to the same antigen later. So these are considered memory cells. Memory cells help us remember antigens, so therefore if we're exposed to it again, our body is ramped up and ready and able to defend us. And then finally, tolerance. Tolerance is the fourth property I want you to know. The immune response ignores self but targets abnormal, right? So that's why autoimmune disorders are so problematic, because most of the time our immune system should be ignoring ourself and attacking foreign things. Can develop over time in response to chronic exposure to an antigen, okay? So exactly what is, we've been saying the word antigen a lot, antibody. Well, let's remember an antigen is a protein or any foreign object that stimulates the immune system to produce antibodies. So here's these antigens on the outside, these antigens producing antibodies. Helping the system, of course, causing the system to produce antibodies. And antibodies are proteins that recognize and bind to antigens. And you can kind of see here that those are drawn like hands to help you understand that that's what they bind to. They bind to antigens, okay? So overview of the immune system. Antigens either infect cells or are processed by phagocytes. So these antigens, bacteria, bacteria, toxin, virus, they are actually engulfed. And then it's literally like, you take a prisoner of war and cut off part of its limbs and stick it on the outside and wave it like, hey, here's the enemy, guys. That's literally, and I think Crash Course gave that example, but that's literally what's happening with this situation. It's called antigenic presentation. So literally they engulf the antigen and then they present parts of it on the outside of the cell. And so this presentation stimulates cell-mediated T cells and antibody-mediated immunity, which would be B cells. So let's look at a picture here. So adaptive immunity, right? Here's your antigens, a virus, a bacterium, another type of virus, different types of viruses. And phagocytes activated, and they present those antigens, parts of those antigens on the outside of the cells, and then that activates T cells, communication and feedback between B cells. So activated B cells give rise to cells that produce antibodies, and so activated T cells find the pathogens and attack them. Antibodies attack by, they stick to it and clump together, help these pathogens be destroyed, and eventually it's the destruction of the antigen or something that's a horrible circle, that we do not want obviously in our body, okay? So antigen recognition. So basically inactive T cells bind to this specific MHC antigen complex. I'm not going to go into that for high school anatomy and physiology, but basically there's this complex right here, and an active T cell binds to it, and they recognize certain classes of it. Well, the T cell will only bind to a complex that contains the antigen that T cell is programmed to detect. And so once they bind to it, they become active. So let's remember a couple of the T cells now. Cytotoxic destroys cells, right? Abnormal or infected cells. So target cells must have specific, proteins on the outside. And then how do they do this? They release, those perforins, which causes pores in the cell membrane. Apoptosis triggers apoptosis, which is cell death. And then disruption of cellular metabolism with certain toxins. So let's see a picture here. You can see activated T cells. Memory T cells are produced. Regulatory T cells provide a delayed suppression. Okay, so they kind of keep everything regulated. And then all those, chemicals can be used to destroy that target cell, whether it's an abnormal cell or a pathogen. So how would an antigen, this is question part 2, how would an antigen on a virus stimulate an immune response, or how would an antigen such as a virus stimulate an immune response, okay? Not only are T cells activated, but B cells are as well. So full activation requires helper T cells. So T cells have to activate a sensitized, which means been exposed to the antigen, B cell. Helper T cells must have been activated by exposure to the same antigen. So you can see that there's some regulation here, some steps you have to go through. And helper T cells bind to the complex, and they secrete cytokines, which are these chemicals that causes the B cell to become activated. Okay, and these B cells become activated to start producing antibodies, at least some of them do. So B cells, cell division, differentiation, antibody production, stimulation by cytokines causes series of cell divisions in B cells. There's two types of daughter cells for these B cells, and you need to know them. Memory B cells and plasma cells. Memory B cells are inactive until a second exposure and then respond by differentiating into plasma cells. So these are what we really remember what's going on, and then they just kind of keep circulating. And then when they're exposed to that antigen again, they activate, they create plasma cells, and your system is ramped up, ready to go to understand what's being infected by to protect you. Plasma cells are activated B cells, and they secrete up to 100 million antibody molecules per hour. That's ridiculous. I don't know if you want to do the quick math in your head. I can't really do it, but that's like crazy amount per second. Quick math. I'll post something, I guess, maybe. But that's crazy. And those antibodies help to defend you, okay? So here's a little graphic here. You have your memory B cells inactive, activated B cell, okay, stimulation, activated B cell. You have so here we're starting here. Active helper T cells activates a B cell, and some of those go into memory B cells, and some of those undergo into active B cells, which turn to plasma cells, and plasma cells produce those antibody molecules. So you definitely can see a multiple choice question there. Which type of cells produce antibody molecules? That would be an easy question for me to ask you, okay? So questions part 3. If you observed a higher than normal number of plasma cells in a sample of lymph, okay, that fluid traveling through the lymph system, would you expect antibody levels in the blood to be higher or lower than normal? Okay, think that through. And so here's some different system molecules here. Cytokines, antibodies, and complement proteins. Again, complement proteins are mostly non-specific, but they do kind of influence antibodies, so I do want to talk to you one more time about it. So cytokines are these molecules. These are really crazy, fancy, collegiate level diagrams. But I do want you to see that basically cytokines are signaling molecules that cause, what do they cause? They cause certain genes to be flipped on or off. They change DNA expression, so therefore they change the function of the cell. There's a receptor gene, so here's the key, gene activation. So how do these cytokine molecules activate B cells? Well, they change DNA expression. And so the B cells are now active because their DNA is being expressed or read differently. Much like if you have a book about cooking and you need to learn how to get season a skillet. I love iron skillets, but they are, you gotta take care of them. But what happens if there's no seasoning? Well, in this huge cookbook that deals with recipes and kitchen maintenance and maintaining your cookware, you would flip to the certain chapter with the cookware, and so DNA expression is similar to that. Antibodies, protein compounds with specific combining sites. So they are attached to specific, remember everything's specific here, it's adaptive immunity, antigens. So they form a humoral immunity or antibody mediated immunity. So antibodies, I love this phrase here, they are molecular watchdogs. They watch and wait for these antigens, viruses, bacteria, other unwelcome visitors. Antibodies circulate in the blood, scrutinizing everything they touch. When they find an unfamiliar foreign object, they bind to its surface, and then a coating of these bound antibodies will be enough to block an infection. However, they are no match for bacteria, so when antibodies bind to bacterial surface, they must act as markers or signals alerting the defensive mechanisms to, hey, this is a bad guy, come destroy it, okay? And so antigen antibiotic complexes may neutralize toxins, clump or agglutinate. Agglutinate is the word for clumping together enemy cells and promote phagocytosis, which is engulfing cells. So this is what an antibody looks like. This is like an RS rendition here. Looks like a Y, okay? There's many different types of classes of antibodies, but I just want you to know the major one, and that's IgG. I don't know if you ever heard of someone getting like, they had like the skin test, and then they started taking shots to decrease their antibody count production in various ways that people who have like really serious allergies sometimes deal with those things. But IgG is the main class of antibodies. Okay, what are they responsible for? They're responsible for resistance against bacteria, and viruses, bacteria, and bacterial toxins. Okay, so exactly how does this all work out in a picture? Okay, so you have your immune system cells here. Antibodies, when they are bound to a pathogen or an antigen, I should say, they can facilitate phagocytosis. So this helps a phagocyte immune system cell ingest it. They can clump together, thus inactivating it. They can activate the complement cascade, which causes, those general non-specific things: inflammation, attraction of white blood cells, cell destruction. Okay. And then, because how does they do that? Well, they could bind to a specific antibody on a certain cell, like those mast cells, and increase inflammation. Okay, so antibodies are really, like I said, are molecular watchdogs. So complement proteins, we already talked about these a little bit, but I just want to briefly review it again. Complement cascade, remember that word cascade? It's a mechanism of action for antibodies. So it causes cell lysis or breaking down or exploding, destruction, by permitting entry of water through a defect created in the plasma membrane. So these common proteins are very important in helping start this whole antibody antigen complex system. So you have the complements, and then these guys, stick these pores in and they destroy them. Again, that is a non-specific, but I did want to bring it up because sometimes, antibodies can activate that system. And then finally, the adaptive response relies on two types of lymphocytes. So let's just review these lymphocytes one more time together. Lymphocytes that mature in the thymus are T cells, and those that mature in the bone marrow are B cells. They have antigen receptors on them, mature B cells, mature T cells. And so this is just another slide that we could review on. T cells come in different types: cytotoxic, helper, suppressor, and memory. Cytotoxic attacks cells, helper enhances antibody production, suppressor inhibits antibody production, and then memory T cells become activated, the same antigen appears in the body again. B cells, though, for our purposes with antibodies, are the main players. Again, they have to be activated by helper Ts, but these plasmacytes or plasma cells produce these antibodies that react with antigens. Okay. Memory B cells become activated, the same antigen appears in a later date. They can activate, become those plasma cells, and produce those 100 million antibodies in an hour. So you can see that when you get an infection, your system, it's very rare for you to get an infection if you have a healthy immune system. And so your system is really built to protect you. So immunocompetent, which means you have a system that works. Immunosuppressed, which means you have a system that is working beneath the threshold of needing to work at. Natural killer cells, remember, they are surveillance cells. They're constantly monitoring. And then you have cell-mediated, which T cells attack directly, and antibody-mediated, which the pathogen is attacked by antibodies produced by B cells. There's the difference there, okay? So the following is a sequence of events involved in the immune response to bacterial antigens. So this is very important because this will be your final question. Macrophages will phagocytize a foreign substance, okay? They will present the antigen on the outside, like, hey, here's the enemy, okay? This is called antigen presentation. T cells begin to divide to produce all these different types of T cells, okay? They begin to specialize. Now, after that, you have that the cytotoxic T cells will kill the bacterial agent directly. Helper T, so here we go. Helper Ts will activate B cells. Oh, sorry, I don't know what's happening. Whoa, gosh, this is crazy. Zooming in, no idea how I'm doing that. Let's just zoom out. Sorry, hey, no idea how that happened, but that was cool. I need to figure out how I did that. B cells will begin producing antibodies, and antibodies will bind to that bacterial antigen. This will attract an army of leukocytes, and these leukocytes will kill the bacteria. You can kind of see that everything has to happen in the correct sequence and the correct order, when the message is being sent. And so there's a lot, there's different ratios and lifespans of these cells, of course, this lymphocyte production happens with stem cells inside of, they get, one set of stem cells will migrate to the thymus gland. That's where they're called T cells, and the other one will stay in the bone and differentiate to form B cells and NK cells. So that is the conclusion of today's lesson. So the final question, part 4, is explain how antibodies are made. So what are the steps that involve antibody production? That will help you really think that through and research that and review. It will really help you understand how antibodies are made. But anyways, I hope this was helpful, guys. Hope you're doing well. Until we talk again, God bless.